You always hear people say, trust the science. But what does that even mean? What is the science? Well, today, we're going to pull back the curtain and see exactly how medical evidence gets made.
You've seen these headlines a million times, right? New drug cures X. This diet will change your life. It's exciting for sure, but how do we really know if it's true? Is it a real breakthrough, or is it just wishful thinking? Well, the answer comes from a branch of science that's basically a global detective agency for our health.
So to get to the bottom of this, we need to talk about epidemiology. It's a bit of a mouthful, I know, but the name literally means the study of what is upon the people. Its whole job is to figure out, what makes us sick and what keeps us healthy.
Okay, so this slide shows a huge leap in tech, but the mission? It's exactly the same. Let me tell you about the OG medical detective, Dr. John Snow. Back in 1854, he had no labs, no computers, just a map of London. During a massive cholera outbreak, he literally went door to door, mapping out who was sick. And you know what he found? It all led back to one single water pump on Broad Street. His solution was brilliant in its simplicity. He took the handle off the pump. That's it. Fast forward to today, and we're using terabyte servers and AI to find those same kinds of patterns. The tools are worlds apart, but the goal, still the same. Find the cause and stop the harm. All right, so how do today's researchers actually hunt for these answers? Well, it really boils down to two main approaches. They can either watch what's happening from the sidelines, or they can jump in and actively do something.
So on one side, you've got watching, or what we call observational studies. You could think of these folks as professional people watchers. They just track groups, collect data, but they never step in. Then, on the other side, you have doing, experimental studies, and this is where it gets interesting. Researchers stop just watching and start, doing something, like giving a new treatment to see what happens.
Now, if we zoom in on those observational studies, there are basically two main flavors. First, you've got cohort studies. These are forward-looking. Researchers grab a group of people, a cohort, who have something in common. Maybe they all work in the same factory, for example. And then they just follow them for years, sometimes decades, just to see who gets sick and who doesn't. Then you have case control studies. These work backward. They start with people who already have a disease and compare them to a group that doesn't, trying to look back in time and figure out what was different, what was the exposure.
And look, these observational studies are great. They're fantastic for spotting clues and potential links. But here's the catch you've probably heard before. Correlation is not causation. Just because two things happen together doesn't mean one caused the other. To prove that a treatment actually causes an effect, you can't just watch. You have to run an experiment.
And that brings us to the absolute gold standard of medical evidence, the randomized controlled trial, or RCT. The way it works is actually pretty simple, but it's incredibly powerful. So step 1, you get a bunch of people who are eligible for the study. Step 2, and this is the magic part, you randomly assign them to different groups. It's like flipping a coin for each person. Step 3, one group gets the shiny new treatment we're testing. The other group, the control group, gets either a placebo, like a sugar pill, or just a standard care that's already out there. And finally, step 4, you just sit back and compare the results. Did the treatment group fare better than the control group? The answer to that simple question, that's some really powerful evidence.
So why is that randomization, that simple coin flip, so important? Why is it the secret sauce? Because it's our #1 weapon against bias. When you randomize, you make it so the two groups are as identical as possible. Not just in the things we can see and measure, like age or gender, but in all the weird hidden stuff we can't measure. Think about it, genetics, lifestyle habits, stuff we don't even know to look for. Randomization just scrambles all of that up between the groups, so the only significant difference left is the treatment itself. It's brilliant.
Okay, so an RCT is super powerful, no doubt, but at the end of the day, it's still just one study. To get an even more solid, more reliable answer, we need to go bigger. We need to combine the power of many studies.
And the way they do that is with something called a meta-analysis. The easiest way to think about it is, it's a study of studies. Seriously, it's a fancy statistical technique that lets researchers take a bunch of different trials that all ask the same question and then pool all their data together into one giant data set. This gives them way more statistical power, and it can help clear things up, especially if the individual studies kind of seem to contradict each other.
So here's why that is so powerful. Picture each individual study having a little bit of random static, or noise, right? Just random chance that might push the results one way or another. Well, when you combine a whole bunch of these studies in a meta-analysis, all that random noise, it tends to just cancel itself out. And what you're left with is a much clearer, much stronger signal of what the treatment is actually doing.
But let's be real, no study is perfect. Even the absolute best-designed, most expensive research can have flaws. So a really big part of understanding medical evidence is learning how to be a bit of a critic, knowing how to spot the potential problems.
Generally speaking, these errors fall into two big buckets. You've got random error, which is pretty much what it sounds like, just the natural, unavoidable variation you get when you study a sample instead of the entire population. But the really dangerous one is systematic error, or what we call bias. This isn't just random noise. This is a flaw baked right into the study's design that's pushing the results in a specific, wrong direction. It can totally tank the whole thing.
One of the classic types of bias is selection bias. This is when the group of people in your study is just fundamentally different from the population you're trying to talk about. And the analogy on the slide says it all, right? It's like trying to find the average height of all adults by only surveying NBA players. Your results are going to be just a little bit off, don't you think?
Okay, another big one to watch out for is confounding. This is a sneaky one. It's when some hidden third-wheel variable is messing with your results. So, for example, let's say a study finds that coffee drinkers have more heart attacks. It's easy to blame the coffee, right? But what if people who drink a lot of coffee are also more likely to be smokers? In that case, smoking is the confounder. It's the hidden factor that's actually causing the heart attacks, not the coffee.
And finally, even with the best design, the statistics themselves can lead you astray in two main ways. On one hand, you've got a type 1 error. Think of this as a false alarm. The study screams, "Hey, this treatment works," when it actually doesn't. It was just a statistical fluke. It's like your smoke alarm going off when you're just making toast. Annoying, but not catastrophic. On the other hand, you have a type 2 error. This is the missed signal. This is when the treatment really does work, but your study was too small or had too much noise to pick it up. This is the smoke alarm that stays dead silent while your kitchen is actually on fire. Much more dangerous.
So, okay, we've got all these different kinds of studies, all these potential flaws and biases. It's a lot. So how do we make sense of it all? How do we know which evidence to trust the most? Well, luckily, there's a framework for that. It's called the evidence pyramid.
Let's build this thing from the ground up, okay? At the very bottom, the weakest level of evidence, you have stuff like expert opinion or basic lab research. These are great for generating ideas, but they're not evidence from actual people. A step up from that, you get case reports and case series. Then we get into the stuff we talked about, the observational studies, like case control and cohort studies, getting stronger now. Above those, you find the powerful randomized controlled trials. And then, sitting right at the very top, the absolute peak of the mountain is the strongest evidence you can get, systematic reviews and meta-analyses, which remember, combine the results of all the best RCTs.
And the rule of thumb here is super simple. The higher you go up that pyramid, the stronger the evidence gets. The research methods are more rigorous, the risk of bias goes down, and our confidence in the results should go way, way up. It's basically a trustometer for medical research.
So what can you do with all this? Here's your personal toolkit for cutting through the noise. Next time you see a headline, ask these 4 things. 1. What kind of study was it? Were they just watching, or was it a real experiment like an RCT? 2. What's the source? Are we talking about one little study, or is this a big systematic review from the top of the pyramid? 3. Think about bias. Who is actually in this study? Who got left out? And finally, 4. Always remember the pyramid. A single study is almost never the final answer. It's just one piece of a giant ongoing puzzle.
So, here's my challenge to you. The next time you read one of those splashy health headlines, you'll be ready. You can look right past the hype and start asking the smart questions. So what's the first question you're going to ask about the evidence?